So I'm Margot Berger. I'm from EGFV ESVV. Today I'm very happy to share with you the work we are doing in our team. I will discuss with you how epigenetics might represent an innovative lever for grapevine breeding in time of climate changes. So genetically identical individuals that can be plants or animals may acquire different phenotypes with time. When I say phenotype, for example, in human, it's like different physical traits. The most known example of that is those of twins. You have like baby twins that can very look alike, but over time, we can notice that they are differentiating in terms of appearance. And this difference is even more enhanced when the lifestyle of both twins are different. Here you have the example of the chromosomes of the twins. Everything that is in yellow is identical, and everything that is in red or green is different. And you can see that over time, these differences are appearing on the chromosomes. However, here we are not noticing genetic differences, it's epigenetic differences. So in a few words, epigenetics, it literally means above genetics, but we define an epigenetic trait as a stably heritable phenotype that results from changes in a chromosome without altering the DNA sequence. So here you have example of natural epivariants. So here, these plants or these animals share the same exact genome. However, they display very different phenotypes, as you can see. And this difference in phenotype are due to epigenetic variation and not DNA mutation. So this is possible because in our cells, all individual has what we call a genome, which is a stored information, and we can compare that to a bookshelf. So you have all this information that is available. This information, thanks to epigenetic marks, will be organized in something that we call the chromatin. It's like an organized information that decide what information will be accessible or not. And this accessibility is also what can allow the gene to be expressed or not. The chromatin can be organized in many different ways, and this is what we call specific epigenomes. So from one stored information, you can have several specific epigenomes, and the existence of these different epigenomes are very important because it's something that the plant needs for its development and also to cope with stress. And the changes in how the chromatin is organized is what will give rise to heritable, developmental, or adaptative traits that can explain how a plant can have different phenotypes. So there is many different epigenetic marks, actually, but in our group, and today I will talk to you about DNA methylation. I won't go too much into detail with that, but what you need to know is that DNA methylation is a very dynamical process that is mediated by specific enzymes. These enzymes are responsible for the addition, the maintenance of DNA methylation over cellular division, and the removal of this DNA methylation. So overall, this process is happening in our cells, and it's very dynamic, and it can be changed by environmental stimuli and stress, for example. So in plants, epigenetics also drives what we call memory. So in a plant, when it suffers stress, it can impact its epigenome, and these changes can be maintained over the plant lifetime. And this is what we call epigenetic memory and somatic memory. And actually, few articles show that this memory in perennial plants are transmitted over years. And that this is what we call the transannual memory of stress. But this memory is not restricted to the life of one plant, and it can be passed to generations. And in plants, we have two types of generation that we can have. The one obtained by sexual reproduction and the one obtained by clonal propagation that can occur naturally for certain plants with marcotage or stolonage. But as for grapevine, the clonal propagation can happen with cuttings, and this memory can pass through cuttings too. And knowing the existence of this epigenetic memory, knowing that the stress that the plant is experiencing can modify epigenetic marks, I will show you a quick presentation of how might be the epigenetic journey of grapevine. So we are working with a grafted plant, meaning that we have two individuals with their own genomes, as a scion and as a rootstock. Studies show that when we graft two partners, there is already something happening between them. There is like a communication between the molecular cues of these partners that can modify their epigenome. Afterwards, these plants are propagated and placed in different vineyards. And each plant will have its own environment and its own life path that can suffer stress for many years, that cannot suffer stress for many years. Each plant can have a different life path, and these different life paths give rise to different epigenomes. So from one individual with one genetic identity, we can obtain, depending on their life path, different epigenome on these plants. And actually, what is very interesting, that some people are beginning to be interested about epigenetic in grapevine, which is very exciting. And there is few articles showing that indeed in grapevine, we are able to see what we can call a terroir effect, showing that a same clone that is placed in two different vineyards can display these changes in epigenetics that can be directly linked to its environment. And in some cases, these differences give rise to what we call phenotypic variability. And this is very interesting, and it's also something that we see naturally in nature. Indeed, in population of plants that display very low genetic diversity, epigenetics variation represents something that can trigger the appearance of phenotypic diversity. And in our group, the question that we are trying to ask is, can we generate this phenotypic diversity in grapevine toward an enhanced stress resilience, and this without altering grapevine genetic information? So to answer that question, the strategy is here. So basically, we want to go from one clone with one genome. And the idea is to create what we will call epivariant population. So plants with same genome, but with many different epigenetic profile. And the idea will be to select on the basis of desirable traits. And in the context of climate change, there is different type of traits that we are interested in, like the better stress resilience, better phenotypic plasticity, delayed phenology, berry quality. But before going to that, the question is, how do we get from this to this epivariant population? And so today I will present you two strategies that we are trying in our group. So it's two different projects that is called Epiclone and Clone Adapt. And to create this epivariant population, we first try to trigger these changes in the epigenome with a chemical treatment. We want to create like a random effect on plants' epigenome. The second strategy is to use the capacity of the plants to remember its life and its past experience to trigger epigenetic modification that is stress-oriented. So first, with the chemical treatment, what we have done is that we have to take in vitro plants. These plants were propagated clonally. And then we treat them with a pharmacological treatment for 30 days. So you have the names of the molecules here. The idea here is to impair how DNA methylation is regulated in the plants. And basically what we are waiting here is to DNA methylation levels to drop, but in a random way. After these 30 days, the plants were put in a normal environment. So we stopped the treatment and we let them for several months developing like that. And then we put them in the greenhouse, waiting for them to grow. And then we were wondering, what is the effect, the long-lasting effect of this treatment on the plants? And what we could see is that we obtained different phenotypes. So some of the plants were higher than the others, some of the plants were smaller. But also what was very interesting, that we see that the metabolic composition of the leaves of these plants was very different, and especially in terms of stilbenes and flavonoids. Of course, we looked at the changes in DNA methylation occurring, and we have global changes in DNA methylation, meaning that we succeeded in creating epivariant for this, from this experiment. So basically, this transient hypomethylating treatment allowed us to impact the metabolic composition of adult plants and trigger methylation differences, and we observed stronger phenotype for some individuals. So now let's see what we obtain with this second strategy, using the stressful life path. And with this strategy, we take one unique mother plant. These plants were propagated clonally, and then we place them in two very contrasted environments. So here you have a very stressful one, like it's a multi-stress. They were placed in a greenhouse with a lower water availability, higher temperature, etc. And this one, the plants were in the optimal conditions. And we maintained the plants like that for 3 years. And what we observed is that the plants that were in stressful conditions displayed an affected phenotype with the reducement of the development and the reducement of the yield. The question that we were asking also in this experiment was, are the plants able to remember their past experience? And to test that, we took part of the plants coming from both greenhouses and we put them in the same greenhouse with optimal condition. And what we were able to see is that the plants coming from a stressful environment partially maintain the phenotype, this affected phenotype, with a reduced development and reduced yield. So now what we wanted to do is to see the impact of these different life paths on DNA methylation for these plants. So first we compare, we looked at the methylation differences between the two contrasted environments, and what we can see is that we trigger differences between them. So the environments impact DNA methylation. And concerning the plants that were in stressful environment and then put in normal environments, we see that we have a partial loss of these differences when they are moved to controlled conditions. So basically here we saw phenotypes due to these life paths that are generated by the condition of growth and that are partially maintained for at least 1 year after changing growing conditions. This growing condition has an impact on methylation landscape, but we lose a part of this epigenetic modification when we move the plants to a controlled condition. So after that, if we took back to our strategy, we can see that both approaches allowed us to create epivariant population. However, before going to this step, we need to answer two questions. The first one is, are these epigenetical changes stable over time and over generation? And the second question is, are these changes related to specific phenotypes that can be interesting? So now, how can we use that to see if epigenetics is a lever in grapevine amelioration? So I will show you the strategy that we plan to use when we will go forward with these experiments. So we want to generate a very big grapevine population with epigenetic diversity. And the idea will be to go very, very deep into the DNA methylation analysis to identify what we call epi-allele, which are very stable changes in epigenetics. And in the meantime, we will screen for desirable traits. So as I said earlier, for example, better stress resilience, delayed phenology, or better quality, for example. And when we will have both of this information, we will be able to look for association between them. The idea will be to identify epi-allele that are linked directly to phenotypic traits that interest us. And in this way, we will be able to obtain grapevine epivariants with desirable phenotypic traits, but without ever changing their genetic information. So I would like to thank you, the member of my group, and especially the professor Philippe Galussi, that is a leader. I would like also to thank all the collaborators that participated in these experiments. I would like to thank my lab, of course, the GFV. I would like to address a special thank you for IVES for inviting me and allowing me to share a little bit of my work. Here you have the review that we have written in 2023 that deals with the subject I presented to you. So if you want more detail, you can read it. And if you want to get in touch and get more information, or if you have any question, you can reach me by scanning this QR code. And of course, I thank you for your attention.